Hi, I'm Richard Sever from Cold Spring Harbor Lab and BioArchive. With me I have David Bartel from the Whitehead Institute at MIT. David, welcome to Cold Spring Harbor.
Thank you, Richard.
You spoke yesterday about microRNAs, small RNAs that regulate the activity of genes post-transcriptionally. Mm-hmm. I want to talk a little bit more about what you said, but one of the things that kind of struck me early on was you made the point that the effects of knocking down these microRNAs could be really debilitating, and we tend to think of them, and people always say, that it's fine-tuning of gene expression. Yeah. So that was kind of a shock to me. Can you say a little bit more about that?
Yeah, I mean, I like the term tuning rather than fine-tuning, because it actually is more of a tuning, and it turns out that these microRNAs do have very striking phenotypes when you knock them out. But even before we knew the knockout phenotypes, we knew that they recognized many targets. And so we know that there are 90 highly conserved families of microRNAs from humans to fish. And each of those 90 have on average about 400 preferentially conserved targets. And that adds up to more than half of the human genes are conserved targets of microRNAs, many on average targeted by 4 or 5 different microRNAs. And so they have this very widespread effect. And it is true that for each microRNA target interaction, that can be a rather subtle, maybe 20% downregulation, sometimes 30, sometimes more. But because they have so many targets, when you knock out the microRNA in mice, which is what many labs have been doing, they've knocked out one or several members of the same family of microRNAs, and when they look at the phenotypes, first of all, for these 90 conserved families, nearly every one of them that's been reported on, where a knockout's been reported on, they certainly do see a phenotype, and they've looked at over half of them and seen phenotypes. And we already know that 15 of these microRNA families, when you knock them out, you have embryonic lethality or perinatal lethality, so that's pretty severe. And there are others that also have very severe phenotypes, like blindness, deafness, infertility, seizures, epilepsy, cancer, etc. So there's a huge range of phenotypes, and many of them very severe from the microRNA. So we know that they're playing very important roles.
And so are you saying in some respects that the severity of the phenotype is not because of the quantitative effect on any given gene, but just because you're hitting a whole bunch of them?
I think that's the easiest way to think about it. Yeah, and in some cases, people have found targets, single individual targets where that 50% downregulation has a dramatic effect, but even there they have hundreds of other high conserved targets that are conserved more than you expect by chance. Right. And so certainly over the course of evolution, we know that biology cares about many more than single targets, and again, each of them regulated by a relatively small amount of tuning.
Right.
So that's kind of our current view of what.
So how does that compare with, for those of us stuck in the 90s, how does that compare with the way we think of transcription factors? Do you think there's not we shouldn't be singling these out as different in the way they work?
I think probably if you look at transcription factors, depending upon the transcription factor, you might also see these minor effects on many targets. It may not be that much different. There are some transcription factors that, of course, will have a 40, 50-fold effects on the transcription of a certain mRNA. But I think more generally, transcription factors might also have widespread and not as effects on the level of microRNAs may not be a lot different for a lot of those interactions.
And the way the microRNAs act is by exerting their effects on messenger RNA.
Yes.
And there's two possibilities there. Stopping its translation and degrading it. And you made the point in your talk that actually it's much more of one than the other.
Yeah. So what we see is that, with one exception, every place where we've looked, over 2/3 of the repression could be explained by the microRNA recruiting factors that deadenylate the RNA and then cause the RNA to get destabilized. And then often over 90% can be explained by this degradation mechanism, leaving somewhere between sometimes 10%, sometimes less, up to 1/3 with this additional mode of repression that's through translational repression, inhibiting presumably translation initiation. So yeah, in general, the majority of the effects that we see are through this mRNA destabilization. The exception, which is really interesting, is that in the early zebrafish, you completely miss what the microRNAs are doing by looking at the changes in mRNA levels. And whereas everywhere else, it's kind of nice. You can perturb the microRNA and be able to know what are the targets and the effects of the microRNAs just by looking at changes in mRNA, which is much easier to do than looking at changes in protein or ribosome protective fragments. And so, but in the zebrafish, that would not work at all. And that was shown. I want to say that was shown by Antonio Giraldez's lab. That in the early zebrafish embryo, that the effects of the microRNAs can only be seen when you look at the changes in ribosome protected fragments.
Right. And is it so that's this early phase of development in zebrafish. Is it early phase of development in other organisms, or is it something unique to a particular lineage?
It's a good question. I think that the same will probably also hold in frogs and in flies, but I'm not sure that people have done those experiments but we do understand what's going on in zebrafish, and we think based on what we see in these other organisms that it'll also hold there. And so what we found in zebrafish was that there's this the effects of the microRNAs, as I kind of said very quickly before, is that they recruit a protein called TNRC6 that in turn recruits the deadenylation complexes, the PAN2, PAN3, or C-server not complex, and that causes the poly A tail to get shorter. And once the tail gets to be a certain length, you get decapping and decay of the mRNA.
Right.
And so what we see in early zebrafish is that the microRNAs cause the tail to get shorter. And in that developmental context, in that regulatory regime, what we find is that mRNAs with short tails are translated much less efficiently than mRNAs with long tails. There's this very strong coupling in the early zebrafish before gastrulation, where short-tailed mRNAs are translated much less efficiently than long-tailed, but what's interesting is that goes away at 6 hours post-fertilization, which is the gastrulation of the fish, and at that point, the microRNA also causes the tail to get shorter. But in that context, a short tail leads to less
degradation.
Degradation, exactly, the degradation of the mRNA.
So sticking with the pre-gastrulation scenario, what do you think, is there a kind of like a priori reason for that? Well, I mean, going back to this kind of tunability or fine tunability, if you're stopping translation rather than degrading the RNA, is that giving you more ability to tune? Is it reversible?
I think you're onto it there. So I'll just add that we see this same sort of transition in translational control in frogs and in flies, and this coupling between the length of the tail and the efficiency of translation, which is very strong in the early embryo of frogs, flies, fish, is also very strong in oocytes, and then goes away around gastrulation. And the reason we think that there is that coupling between the length of the tail and the efficiency of translation is that this is a time in development when, at least up until about 3 hours post-fertilization, this is a time in development in which there is no transcription.
Mm-hmm.
Okay, so the mRNA is all maternally inherited, and yet the embryo cells still need a way to regulate genes. They can't do it by transcription, and the way they do it is by changing the tail length.
Right.
Okay, so they'll have this phenomenon called cytoplasmic polyadenylation, which will extend the length of the tail, and that will cause a massive increase in translation.
Uh-huh.
And then they also have other ways of shortening the tail. And so they can adjust the amount of protein output.
So do you get an oscillation there?
Yeah, you can get, depending upon the gene, you can just get different output at different points in early development through this polyadenylation mechanism.
Right.
And then so then the question is, well, why does it go away?
Right.
And the reason we think it goes away is that later in development, transcription has already started up. And that's a great way to regulate genes.
Right.
And so you don't need this tail length control to regulate translational output. And instead, what the cells use is transcription, and they also use mRNA stability.
Right.
Okay, that's what the microRNAs are doing. They're changing the stability of the mRNAs, and that's what we see in like I said earlier, in all these post-embryonic contexts. And so when the cell is using mRNA stability to regulate genes, then it's not such a great strategy to only translate the mRNAs with long tails, because those are the mRNAs that just came into the cytoplasm.
Right.
Whereas the RNAs that have been there a long time have shorter tails, and the cell would not want to discriminate against it. It wouldn't want to do this age discrimination against the older mRNAs if it's had those RNAs stable. It wants to use them. It wants to use the RNAs with short tails just as much as it wants to use those with long tails, if it's using mRNA stability as a mechanism for gene regulation. Right. So when you use, the idea is that when the cell switches over to this mechanism of using mRNA stability to regulate genes, which they use a lot, they have these massive differences in mRNA stability, then the cells switch away from controlling translation based on the length of the tail.
Right, right. And so one of the things that I thought was very interesting about your talk was when you were looking at the length of the poly A tails, you were discriminating between steady state levels and sort of a better experimental approach to make sure you know what the length is and when. Can you tell us a little bit about that?
Sure. So what I described so far was in the early zebrafish embryo, and there, it's not really a steady state situation. You have development happening, and you can see these tail length differences very readily from the microRNAs. But interestingly, what we found, if you just look in mouse 3T3 cell, the fibroblast cells of mouse, and you're just growing cells at steady state, you have a microRNA that you've induced that is there at very high levels. And then you just look at the tail lengths, the mRNA poly A tail lengths of the targets of that microRNA. Surprisingly, at steady state, you see no difference. The distribution of tail lengths is the same, whether or not the mRNA is a target of the microRNA or not. And that caused us to realize that we shouldn't really be looking at steady state measurements. We need to look at pre-steady state. We need to go and just look at what's happening to the newly transcribed mRNAs and beyond, and get measurements of the dynamics that you can't get at steady state.
So what's the timeline that you're thinking about then when discriminating the point at which steady state becomes a reasonable measure? Or
I think at about 8 hours, things have pretty much approached steady state in these cells. There aren't that many mRNAs that have half-lives much longer than that. There's some, but you're pretty much approached steady state by 8 hours. So we'll do a metabolic labeling time course, that'll take a very early time point and some intermediate ones, and then 8 hours, and then just isolate the RNA that was just transcribed over those time intervals and measure the tails.
Uh-huh.
And then when we do that, we can see there's very clear differences in the microRNA effect. We can see that the microRNAs are clearly shortening the tails, which we can see at these intermediate time points.
And when the tails shorten, is there like a threshold at which you're basically done? You're going to get degraded. What's the we set out to do these experiments to look at this microRNA effect, but we can look at these more general principles of tail length shortening, deadenylation rates, and then the rate of decay once the mRNA gets to be a short tail length. And so we can get these measurements, which we knew for a very small set of genes previously. So people in the 90s, like Anbin Shu's lab and others in mammalian cells and Roy Parker's lab in yeast. In mammalian cells, we knew from those early experiments that the tail length dynamics and mRNA decay dynamics that are associated with tail length for 4 mRNAs. Uh-huh.
And when we realized that we had the data sets now to be able to look at this for thousands of mRNAs, that was very exciting to us. And so now we can use these metabolic labeling data sets to model for close to 3,000 mRNAs from 3,000 different genes. We can model the initial tail length when the mRNA goes into the cytoplasm. What is the initial time length at that early point, tail length at that early point, and then how rapidly did the tails get shorter, and then once they get shorter, how rapidly are they degraded? Okay, and we get that information now for close to 3,000 mRNAs from 3,000 genes.
And so what's the sort of is there a broad spectrum of behavior? Do they all look the same? Does this all happen at 30 nucleotides?
Yeah, good. So the what we see is that the deadenylation rates for mRNAs from different genes can vary widely, 1,000-fold. So there's some mRNAs that are deadenylating at 30 nucleotides per minute. Okay. And there are others where the tail is shortening at 0.03 nucleotides per minute, like 1 to 2 nucleotides per hour.
Right.
Okay, on average for that.
So a couple of orders of magnitude.
So that's 1,000-fold and 1,000-fold difference. And so that's one thing we find. And then what's interesting is that once the tails reach a short length, the rate of decay can also vary.
Right.
Okay, and so what we see is that can also vary 1,000-fold. So the short-tailed mRNAs from a gene where the tails were getting where the mRNAs, their tails were getting shorter more rapidly. Once they reach a short tail length, the mRNA decays more rapidly. But if the tail is deadenylating more slowly, once they reach short tail, they decay much more slowly. And so this is very interesting to us, and what it does is it prevents the mRNAs that have very rapid deadenylation from accumulating as just staying there in the cell with very short tails. Otherwise, you could have this big buildup of very short-tailed mRNAs. So we just don't see that. And so this explains why. It's because for the mRNAs where their tails get shorter more rapidly, they also then decay more rapidly once they reach a short tail length. And that allows this huge differences in deadenylation rate, this 1,000-fold difference, to impart a similarly large difference in decay rate.
Right.
And so you can have a really broad spectrum of post-transcriptional behavior for mRNAs from different genes, and of course that's to go with transcription and other things is what gives us the regulation that we have.
Well, it sounds like a good advertisement for looking hard and quantifying things.
Yeah.
So, well, it's been fascinating talking with you. Thanks very much, and
appreciate the opportunity.
And good luck in the future.
Thank you.
Thanks.